Reproducibility is important in science. Neuroimaging is not an exception. There are many discussions and debates about the reproducibility in neuroimaging. But here, I'm going to only limit it to three perspectives that are related to common practice and modeling in neuroimaging. So the first perspective is about statistic. The typical practice is to focus on statistic only without reporting effect size nor uncertainty. The second perspective is filtering. So the common practice is to filter the results per p-value of 0.05. Only report the part that survives the thresholding, then hide the rest. That automatically creates biases. The third perspective is modeling. So the dominant approach is called massively univariate analysis. With that approach, basically you build as many models as the number of voxels, regions, or matrix elements. The automatically penalty is multiple comparisons. Everybody knows how heavy the penalty is. So I'm here to offer an alternative using Bayesian multi-level model. So we build one model, so automatically the multiple comparison issue is dissolved. More importantly, that also we can improve the focusing. We report full results without filtering, and also we show effect estimates and their uncertainty. So let's start with the first perspective. So let me ask a question. What is the distance between Earth and Moon? If I answer that question with t-statistic value of 4.25 or p-value of 0.01, would you be satisfied? If you consider that's ridiculous, just examine the results in the literature, attend presentations or poster session. You know what I'm talking about. So back to the original question, the distance between Earth and the Moon average is almost 400,000 kilometers. But that's not good enough because depending on the relative distance locations of the two bodies, there's some uncertain range. So we better provide that range. So back to the focus of a t-statistic value. So with that value, there's only two pieces of information at most. The first piece is directionality. That's you either have the positive or negative sign. In addition to that, you have sort of the strength of the statistical evidence. But in science, that kind of information is not as informative as the full results, and we consider such a theory is immature. So there's a lot of room for improvement. Basically, we can report the effect size and their uncertainty. Better yet, we can present even full distribution of each effect. So the second perspective is about the filtering results. So the traditional framework is null hypothesis significance testing. We start with the assumption, we pretend that there's no effect at all everywhere in the brain. Then we started a courageous fight against the p-value of 0.5. In the end, either you win or you lose. So in the end, you filter the results, then hide the part that you don't consider the evidence that is weak. So there are many issues, too many to list. Here, I briefly just mentioned a couple of them. So automatically, we artificially separate the results with two. One, we consider sort of truth, the other one is falsehood. And it's pretty much like we draw a line in the sand. In reality, what's the difference between the two sides? Most of the time, they're not necessarily big. So we artificially basically create some biases, distorted information, and in the end, that's not going to help in reproducibility. So let's combine both perspectives. Let me use an example. Suppose we have a research hypothesis about one effect that we suspect would be positive in one particular brain region. So suppose we have 11 teams, they design their own experiment to validate the hypothesis. Or we have one same data set, ask 11 teams to analyze the same data. Suppose this is the results they came up with, right? So among those 11 results, only 4 of them managed to survive the so-called p-value of 0.05. So in publications, you would only see those 4 results. Typically in neuroimaging, there's no effect size, no uncertainty. So 4 out of 11. Do we consider that's cherry picking? And it's from a meta-analysis perspective, you cannot do much about it other than just based on the so-called coordinates-based approach that is considered crude. So what can we do better than that? Well, I think we can. So if we don't do any censorship, suppose we have all the 11 results available, not just limited to those lucky 4 ones. So with those 11 results, suppose we arrange them based on their mean effective size from left to the right. Among those 11, clearly 10 of them, their average value is greater than 0. So they're sort of largely consistent with our original research hypothesis. More importantly, we can reach some agreement among those 11 studies or results, or we apply some democratic process. So we build a hierarchical model, incorporate all the information available, which suppose we have the effect size plus their uncertainty, then the agreement is horizontal blue line plus the uncertainty range. That result is definitely more convincing and robust than the traditional approach that only allows those 4 lucky ones, filtered ones. So from meta-analysis approach, this is also much better than the coordinates-based one. So let's switch to the third perspective, modeling. So the dominant approach is massively univariate analysis. With that approach, again, we build many models as the number of voxels, regions, or matrix elements. No information is shared across the whole brain or across all the elements. I'm not referring to the voxels among the neighboring voxels in the brain, because that's usually sort of correct during the so-called multiple comparison correction. I mean, talk about the remote regions. They are not dispersed all over the place. If you're really interested in, you can plot the distribution of those brain regions. I think everybody would agree they would be tend to be Gaussian distributed. So, but the problem with the traditional approach is, in the end, we have to suffer from the correction process. There are many problems with that. For example, the penalty is heavy, small regions get penalized and discriminated. Then we have to apply the filtering threshold, P0.5. No uncertainty information available. Cultures don't respect, not necessarily respect the anatomic regions. So let's approve that, provide an alternative. So we build just one model. That model incorporates all the regions in the brain. So automatically, no more multiple comparison issue, and that would allow us to report full results and estimate effect size uncertainty. And more importantly, we can also validate the outer model. Let's use one particular example to demonstrate the modeling approach. Suppose we have 124 subjects at the population level. So we have one experimental variable. So the traditional approach, if we have 21 RIs, basically we build 21 general linear model. In this case, just a simple regression. So in the end, we have the multiple comparison issue, right? So alternatively, we just build one model at the bottom. So that model, as you can see, there are 2 indexes. So index I codes the subjects. Index J represents those 21 RIs. So among those 20 RIs, I mean, the one model framework, you can see there's intercept plus the slope effect. That's at the population level, shared by all the RIs and subjects. In addition to that each subject has their own component, and they have each RI has their component too. So that's basically use this one framework to decompose each effect. So with that, in that time, modeling framework, we basically constrain all those RIs with a Gaussian distribution. That constraint does not come from whatever we specify. We just say the distribution is Gaussian, let the data to decide how that the strength of that constraint. In other words, the variance of that distribution is totally decided by the data. So in that sense, the traditional approach, the massive univariate generator model, basically is a special case in the sense that the variance is infinity. That means no constraint. So with this model, we solve it, then we get the results. Let's see what we can get out of the model. So on the right-hand side, you see those among those 20 RIs, we show the posterior density plot, the histogram or the distribution of each effect. On the right-hand side of each posterior distribution, you can see the number. That number basically shows the statistical evidence. The green vertical line is the zero effect. So the evidence is the area under the curve on the right-hand side of the green line. So that's basically shows the statistical evidence. So you can present all the 21 regions without filtering, no hiding. So the effect size is there, uncertainty is there. So you can highlight some of them, but you don't need to hide the risk, even if the evidence is weak. So we don't stop there. There are more we can do with the model approach. First of all, we can validate the model from different ways. The first is we can use the leave one out information criteria. So if you're familiar with that, you can see those values clearly shows that the Bayesian approach is way much better, but we have a better approach. It's more intuitive. So in those two plots, they have the same black curve. That is basically the original data. So now we have two models. Let's see which model performs better. We just use each model to generate new data. So use the model to generate similar data. The data is the blue curve. So that you can see, compare the two models. On the right-hand side, the model tends to fit the original data way much better than the traditional approach, the massively univariate generator model. So with that, I want to summarize my presentation. So we offer three better perspectives. The first, I suppose we can focus on the full results, the estimation and their uncertainty. We don't really have to focus on the statistical value. Second of all, we can present the full results. You can highlight them, some of them, but you don't have highlight the rest. Don't create some artificial bias. So the third perspective, we can improve the modeling by integrating all the information, provide some calibration process that will achieve higher efficiency. So lastly, I would just want to say, contrary to the public impression, Bayesian modeling is not about the subjectivity. Instead, we adopt the approach to more efficiently to integrate the information and calibrate the information, present a more powerful tool. Thank you.
Okay, let's get started. So this is Chen from University of Maryland, School of Medicine, Division of Biostatistics and Bioinformatics. And today I would like to share with you about our recent progress in an interesting study called the Empirical Bayes Estimate of Primary Threshold in Cluster-Wise fMRI Inferences. This is a joint work with my PhD student, Yunjiang Ge, who is the leading author, Stephanie Hare, Peter Kochanov, Elliot Holm from University of Maryland. And the topic of this talk mainly focused on a very famous and long-time standing problem of multiple testing in fMRI data analysis. This could be brain activation analysis to some external stimuli measured by task fMRI or seed-voxel-based brain connectivity map analysis at the group level. Suppose we have capital V voxels and V hypothesis test can be performed simultaneously And the p-values and z-test statistics can be calculated by general linear model or multivariate regression that account for the dependencies between the voxels. And the goal usually is to select a subset of voxels that is significant associated with the experimental condition or the clinical status of a subject, so across different groups. And we want the selection of the subset voxels with high sensitivity, essentially with high power and a low discovery rate. In that, we have high replicability and reproducibility. And as we know, the conventional multiple testing correction strategies may not suit for the multiple testing in a 3D brain image because they don't take into account the spatial constraints of super threshold voxels. And usually, so there are several methods has been developed, and the most commonly used method is still the two-step cluster-wise inference procedure, which is popular because the sensitivity or power is high. So usually in the first step is a screening step. All voxels is just you screening all voxels by a pre-specified cutoff with a, we call that primary threshold. For example, p-value is less than 0.001. If your one voxel's p-value is less than that, you keep the voxel. Otherwise, you make it 0. And then at the second step, you can perform permutation tests on connected clusters. So essentially, if a set of voxels are adjacent to each other, you consider as a cluster. So you can do the cluster-wise, family-wise error rate control by doing the permutation test. And the reason that the two-step procedure provides a better balance than the one-step FDR method or all types of multiple testing, traditional multiple testing methods, is because this is a kind of provides double level of FDR. So in the first step, essentially it's a binarization. The threshold usually is much less stringent than FDR or buffer warning correction to ensure the high power. And after the first step binarization, all the points in the 3D brain space can be considered as a binary point process. And at that level, because in first level, the cutoff is much loose, and at the second level, there are many false positive voxels. And the second step, because there's underlying assumption that the false positive voxels, they don't appear that they don't appear in a large cluster. So false positive voxels usually just appear more independently, unconnected. So by using the combinatorics probability, they further control the FDR. So they do the FDR in two steps. And on the other side, the true positive points are more likely to be connected into a large cluster. So the power is not reduced much. And the FDR at two steps, you stack them together, and the overall FDR is better controlled. For this procedure, the primary threshold selection is critical because over conservative cutoff can reduce the power or sensitivity and lead to no positive findings. And so because too few true positive points can be kept and then form a cluster, and then the permutation test will select that. If you choose a too loose cutoff, can produce many false positive, and because there are so many false positive, the false positive can be connected into large clusters. And because it's a false positive, you are very unlikely to see that again, different study in similar experimental settings. there's famous paper in 2014 by Wu and the ETL. So they suggest a cutoff them 0.001, and quickly this cutoff has been well accepted by the community and for all studies. And in this study, we just consider that essentially for different studies, they could have different effect size, different sample size, different noise level, and a lot of different settings from one study to another study. And we hope we can make the cutoff primary threshold more adaptively than a predetermined level, so more data-driven. And we propose empirical based adaptive threshold selection. We call it EBAS for the primary threshold, optimal primary threshold selection. So first, we want to define what is optimal. Generally, the optimality refers to when you can control your FDR at the level you specified. For example, FDR as 20%. The optimality refers that at that level, you can reach the maximum power. And so if I write that mathematically, so it's a cutoff theta is optimal that you can just, if you constrain the overall FDR is less than alpha, then the cutoff can maximize your sensitivity. So the sensitivity is determined by the theta, and the FDR is determined by the theta. Theta is the primary threshold. So the true, the sensitivity is essentially the true positive voxels that pass the threshold over the overall how many true positive voxels. And the FDR is just all voxels pass the thresholds, how many of them are false positives. However, these two are unknown. So without any information of these two criteria, it's almost impossible to optimize the cutoff. So instead of just choosing predetermined cutoff, we resort to a empirical based method, which is well established actually. So due to the nature of binarization of multiple tests, you set a set to 0 and set a set to 1 by the screening procedure. We consider the test statistics from all voxels can follow mixture distribution and exactly two component mixture distribution. So for each voxel, the test statistic essentially is coming from a mixture distribution. And the pi 0, which is the kind of pi 0, f0, and the pi 1 and f1 are the two are the priors. So essentially, pi 0 is the proportion of the voxels coming from the non-distribution, and pi 1 is the total number of total proportion of voxels coming from the non-distribution. Usually, pi 0 is much greater than pi 1. For example, pi 1 equals 10%, and pi 0 equals 90%. And the non-distribution is f0z, and the non is f1z. So essentially, you can see the green curve is now f0, and the purple one is f1. So so you can say it's only a small proportion of the voxels you can consider that is coming from the non-distribution. And so essentially for different effect size, different noise level, and different sample sizes, the distribution of f1, f0, pi 0, and pi 1 will be different. And accordingly, we can estimate this pi 0, pi 1, f1, f0 by using an empirical based method. And there has been many algorithms to calculate that. by Efron's work and later by Wu's work in 2006. So essentially, we can estimate pi 1, pi 0, f1 based on the distribution of the data. So the posterior distribution of the non distribution probability is essentially coming from the data. And then based on the data, we can estimate for each cutoff, we can estimate the estimated sensitivity and FDR. So that's a kind of a good property that even though we don't know the true sensitivity given the cutoff or FDR given the primary threshold, we can use the empirical base estimated sensitivity and FDR and provide a good estimate. So that's that provides some input data or some conditions to optimize our primary threshold estimation. So specifically, we propose an objective function that essentially is kind of combines true positive range and sensitivity. So essentially, we want to select a threshold that can maximize sensitivity that ensure that the true positive discovery rate is high, or in the other words, the false discovery rate for cluster-wise inference is below the preset alpha level. So so the zeta is the optimal solution for the primary threshold based on the estimated, empirically based estimated sensitivity and FDR. And although sensitivity and FDR are estimated based on the marginal statistics, the integration will provide a good estimate of the true value of the sensitivity and FDR. So we have some theory to prove that. And to implement the algorithm, essentially there are three steps. So first, we need to do the empirical base estimate and calculate the sensitivity and FDR, and then we identify the all support for Z theta. So essentially, we select the range of theta that to ensure that all false positive voxels in the 3D space by permutation test with considering the spatial coherence that there won't be a large cluster can be detected by permutation test. So so we develop a new algorithm to consider how much noise can, for example, if you choose a cutoff like here, you need to consider such amount of false positive voxels as a proportion. And then if you put those proportions of false positive voxels in the 3D brain, how likely they will be clustered to a large cluster. For example, the cluster size equal to 10 or 20, and then to be positive. The 10 or 20 is determined by the proportion of the true positive clusters. And then if you have more true positive clusters, then the cutoff size is larger. So you determine what's the Z support of Z theta you can choose. And then we do a cost-sensitive classification algorithm to determine what is best choice of Z theta. The calculation for that is pretty fast. So by that standard, essentially we choose optimal primary threshold that maximize the sensitivity with well-controlled cluster level FDR. So we have some theory developed, and we'll post the theorem proof on the bioarchive that the threshold Z is consistent estimator. And the first is the consistent theorem. The second is so optimality. So basically, for all cutoff that satisfy that the FDR cluster FDR is less than alpha, our selected primary threshold using the objective function will give you the maximum sensitivity. So we examine these examples using motivating data. So essentially, we just have 98 schizophrenia patients and 107 controls with matched ages and sexes, and resting state fMRI recorded, and the primary variable of interest is a psychosis score, and the and then the Seyedeh-Rezvan method is used with the default mode centered Seyedeh-Rezvans and with a 10 millimeter spherical size. And then the total number of voxels is 215,348 voxels. And we can compare our method to several other traditional methods as the primary threshold selection. For example, including the p less than 0.01 FDR and the bufferonic correction. And using this data set, we found that there is no positive findings by using the bufferonic correction or FDR, only because both of them are too stringent, and only p less than 0.001, and our method have the positive findings. So once again, we can say that the traditional methods, FDR and the bufferonic correction, won't work because the power is limited. And to compare the two methods, we essentially apply the EBAS method to better observe that. We can just have the estimated F0 and F1, and the purple line is the cutoff of EBAS method, and the 0.001 is Wu's papers predetermined cutoff. And you can see, if you consider this is the true underlying true positive proportion, clearly there is a two component in the mixture distribution. And if we choose cutoff like here, and there certainly sacrifice a certain bound of false positive, but the power, the proportion for true positive is included more. So there's always a trade-off between the power and the false discovery rate. And this is 0.001. And if you choose a too stringent cutoff, you won't have any results. And then we do the experiment actually is we randomly select a subset of 30 subjects of with disease and 30 healthy control subjects. And then for the study B, we just using the 60 subjects randomly selected from per group. And then for C, we essentially choose original sample size. You can see that for the second 60 subjects per arm, there's no significant results by using the p-value as 0.001 predetermined cutoff. And for the our method, we review kind of positive findings that is very similar to the original data set. So essentially, by theory, we should observe pretty consistent results because the sample size increasing and the data should be very similar of the effect size should be very similar of the each subsample size. And then we should observe gradually increasing clusters of positive findings after multiple test control. And we do see this by e-bias, but if we do the predetermined p less than 0.01 method at a certain level, we don't see any positive findings. So in that case, if we consider the using the death index between these two and these two, the value will be just 0. That is essentially to say that the reproducibility or replicability is lower by using the predetermined cutoff and using the empirical based method and use the data structure, data-driven method that can increase the reproducibility. And so here is the different views, the corner view and the sagittal view of the two methods. And also we did a simulation study that with the underlying truth, this is just a two group test. And then with two group test, if you have original graph and then you have different sample size, then this your test statistics. And we also have smooth data at the different smooth levels. And under each setting, we did 100 times experiments, and we summarize the results. And so essentially, you can see from the table that for all settings, the true positive rate essentially is the sensitivity, and the true discovery rate is, we say, it's 1 minus the false discovery rate. So essentially, you can see that all the other methods, essentially, you don't see any true positive findings. So the sensitivity is very low, and the e-bias is more adaptive, and then you can find more true positive findings. And no matter what is smooth level, sample size, effect size, you have similar trends that by using the e-bias, your power is mainly focused around 70%, and the false discovery rate is around 25%. And so there's no free lunch essentially if you use a single value for primary threshold cutoff. This is always a sacrifice between the false discovery rate and the power. But you can see by losing a little bit false discovery rate, we increase power a lot. So we consider that's a good balance, because the replicability and the reproducibility depends on two things. One is the sensitivity, the other is the false discovery rate. And if we can balance, we consider as risk range, there will be a better balance, and then the reproducibility would be higher, so we can have better findings. So the conclusion is that the primary threshold in two-step cluster-wise inference is critical. And rather than using a predetermined cutoff, we propose a data-driven empirical-based method to optimize the primary threshold. And the optimized cutoff is can be calculated by the sensitivity and FDR using the base estimated values. And the results that show that e-bias can provide a better balance for the trade-off and improve the replicability of findings. So better balance is increased power with a very small sacrifice of a false discovery rate. So that's pretty much of the talk. Thank you very much for listening to this talk. And yeah.
Hi, everyone. Thank you for being here today. I'm Mejia from Indiana University, and today I'm going to tell you about template ICA, which is a method to produce reliable subject-level brain networks through big data priors. So it goes without saying almost that in 2020, there's a vast wealth of fMRI data available through data sets like the Human Connectome Project and the UK Biobank, as well as more specialized data sets like the Baby Connectome Project. So the idea we wanted to pursue here was to use information encoded in these big data sets through templates. So templates are empirical population priors, and they describe the distribution of an effect in a population. So here I'm showing the mean value across a population of an attention network, as well as the variance between subjects in that population of that same network. These can be estimated using big fMRI data sets or using holdout data for a more specialized template. They can then be used in a Bayesian model to improve estimation of the same effect in a separate data set. And here I'm going to be specifically talking about a Bayesian ICA model. So before I present that model, I want to just give a brief review of single subject ICA. So we want to decompose fMRI data in the matrix Y into two matrices A and S. And S here contains the independent components. So each row of S represents a functional brain network or a source of noise. And A here includes a time series associated with each independent component. So the limitations of single subject ICA are pretty well recognized, and these include low signal to noise ratio in most single subject fMRI data sets, resulting in noisy estimates of the independent components in S. It's also difficult, if not impossible, to match independent components across subjects or studies. And so what we typically do is we start with the group and then we go to the individual. So we first pool subjects in a group ICA, and then we have a number of options to obtain subject level brain network estimates from those group ICs. So we could use dual regression or back reconstruction. These are convenient and fast, but ad hoc approaches, and they may produce noisier results. We could also use existing optimization-based approaches like group information guided ICA. These seek to maximize similarity between subject and group ICs and therefore provide a matching. So what we're proposing is template ICA, which is a Bayesian statistical framework. And one of the major differences between this and existing methods is that template ICA incorporates the variance of the ICs as well as the mean represented by the group ICs. And this provides optimal estimation of the subject level ICs. And because it's a statistical framework, it can be used to perform principal thresholding for visualization or quantification of the size of the networks through a inference procedure. So I'll just give a very brief overview of the template ICA framework. So the goal here is to inform single subject ICA through population priors. So we start with the same single subject ICA model I showed earlier, and we just add one additional layer. And in that second layer, the subject ICs in S are modeled as being related to the known group ICs in SO through a deviation delta. And we assume that those deviations delta are normally distributed with a known variance. And so this known mean and variance is known as the template. And if you'd like to learn more about this model, I encourage you to check out the reference that's provided here at the bottom. So we could also go one step further and incorporate spatial priors on the subject level deviations to account for the fact that those deviations tend to be smooth. So we just have to change the prior structure somewhat, and in particular, we include a spatial correlation matrix, R, which models the relationship between neighboring vertices along the cortical surface. So again, if you'd like to learn more about this particular model, I encourage you to check out this other reference that I'm providing here at the bottom. So now I'll just show some example results and a brief reliability analysis. We estimated a template based on data from the Human Connectome Project 500 subject release. We chose 16 brain networks for the template from the 25 component ICA from the HCP. And we estimated the mean and the between subject variance of each of those networks based on resting state data from 461 subjects. And I'm showing an example IC which represents an attention network here. So at the top, you can see the population mean of this IC, and at the bottom, you see the between subject variance. And one thing that you may notice is that the variance is showing somewhat similar patterns to the mean. So in particular, you see that the areas that are most engaged in this network on average are also the areas where subjects tend to differ the most. And these differences could be due to topological differences or differences in intensity or connectivity within that network. One thing that's important to point out here is that these priors are spatially varying. So the variance across subjects is different in some areas. So it's higher in some areas and it's very low in other areas. And this is important because it gives flexibility to the model. So the data is allowed to speak louder where subjects are known to differ from each other. So in those areas, the model is most flexible. And in areas where subjects are known to be similar, the models provide more shrinkage. So this provides a very flexible framework where the data is really allowed to speak while benefiting from the prior. So here's some example IC estimates from one test subject, and this is 15 minutes of data. And I'm showing estimates based on dual regression, template ICA, and template ICA with the spatial priors. And you'll notice probably that the template ICA estimates are substantially smoother than those produced by dual regression. At the bottom here, I'm also showing areas of engagement that we identify using the template ICA framework with or without spatial priors. And this is based on an inference procedure. So this is not available through dual regression because it's not just the threshold of the intensities, it's actually based on a statistical inference procedure. So we can also produce what we're calling subject effects using template ICA. So these effects are those deviations from the model, so the differences between the subject and the group ICs. And so you'll notice some hot spots in bright yellow, and these represent areas where the subject is more engaged in this network relative to the population. You'll also see a couple of cool spots in green or turquoise, and those represent areas where the subject is less engaged for this particular network relative to the population. And again, because this is a statistical framework, we can perform inference on these maps and identify areas of significant positive or negative deviation. So I'm showing those maps here on the bottom. One thing you may notice here is that the model with spatial priors has larger areas of deviation. And this is because by including the spatial priors, we have better power to detect true effects. In this case, the deviations of the subject from the group. And so these thresholded maps may be useful for visualization and interpretation, but they also may be useful for quantification of the differences of a subject from a population. Okay, so I just want to show some very brief reliability study results. So we did scan-rescan reliability based on the interclass correlation coefficient. So this, on the y-axis, we're showing the interclass correlation coefficient, or ICC, and that represents the percentage of reliable subject level information in an estimate. So the lines here represent those 16 networks from the template, and I've colored them based on the type of network they represent. So there's 16 lines here. And we varied scan duration at each visit from 5 minutes to 15 minutes. And one thing you'll notice very easily is that template ICA outperforms dual regression regardless of scan duration, and to a very similar degree across scan durations. But one thing that you may also notice is that if you compare the reliability of the template ICA estimates based on 5 minutes of data with the dual regression estimates based on 15 minutes of data, template ICA has much stronger reliability than dual regression does, even with 3 times of the data being used in the dual regression estimation. So one thing that this illustrates is that while it's always better to get more data, it's also very important to choose an optimal, whenever possible, an optimal estimation strategy. So template ICA is able to make the most of a limited amount of data, and it also shows an improvement for longer scans as well. So just to recap, template ICA leverages information in big fMRI data sets through empirical population priors. It, as we've seen, produces smooth and reliable subject-level brain networks and subject effect maps. It can also be used to provide principled thresholded maps for quantification and visualization. And it's also computationally efficient. The model can be estimated in a matter of a few minutes. So the template ICA method is implemented in both R and MATLAB through the R Bayes fMRI package and the MATLAB template ICA toolbox. So feel free to check those out if you're interested. And with that, I'd love to thank all of my wonderful collaborators as well as my students. and encourage you to check out our posters later in the conference. Thank you so much.
Hi, I'm Rezvan, and I'm going to be talking about a hierarchical Bayesian model that we've been working on to get individual and population level resting state networks from very large scale data sets such as UK Biobank. In recent years, there has been growing interest and efforts into getting single subject models of RSNs, and available models often either start from a group model and estimate single subjects based off that group model, or they start from single subject modeling and combine those to get a consensus group model. More recently, however, there has been also some efforts into getting hierarchical models that allow for bidirectional flow of information between subject and group, such that the accuracy of estimations at both levels will be improved. And one such model is the framework of probabilistic functional modes, or PROFUMO, that Sam Harrison and colleagues proposed a few years ago. In this study, we wanted to continue on that work and get a hierarchical bidirectional model of RSNs, but we also want our model to be scalable to very large population data sets, such as UK Biobank, that is expected to have 100,000 subjects. And as it can be expected for such large population data sets to include heterogeneous populations, we want our model to be able to accommodate heterogeneity in the data as well. For that purpose, we propose an extension to PROFUMO, and our model is based on a stochastic variational Bayesian inference. Before I go into the detail of the proposed model and results, I'm just going to make a quick dive into the PROFUMO and its utilities. So PROFUMO is a variational Bayesian framework that infers RSNs hierarchically, meaning that it allows for both top-down regularization of the RSN parameters from using group-level priors, and also bottom-up regularization of the parameters using single-subject decomposition. And by iterating between group-level and subject-level estimation, the model continues until convergence. PROFUMO is hierarchical in two senses. One is over the spatial layout of the RSNs, and the other is over the functional connectome between the time courses of the RSNs. In addition to these two key features, PROFUMO also relaxes the assumption of a spatial and temporal independence among the RSNs, which has been a key assumption for ICA type models. And therefore, it provides a powerful and flexible framework to infer RSNs based on fMRI data. Okay, once we have the RSNs estimated, it is often of interest to find functional connectivity between the time courses of RSNs and build a functional connectome. And one prominent approach to do that is to run group-level spatial ICA followed by dual regression. However, recent works by Janine Bisterbosch, Sam Harrison and colleagues have proposed that functional connectivity estimation in this paradigm might be compromised because firstly, these cross-subject topological variations are not explicitly accounted for, which can instead result in biased estimations of functional connectivity. Additionally, because the model has an assumption of a spatial independence between the modes, it can fail to capture true spatial overlaps between them, which also results in biased estimations of functional connectivity. The above references have shown that PROFUMO can ameliorate both these problems and yield more interpretable measures of functional connectivity. Right, so the model is good and useful. However, if we want to apply it to very large data sets, thousands of subjects from UK Biobank, for example, we're going to start needing tens or even hundreds of terabytes of computer RAM memories, which is something that is computationally unavailable at the moment, and we can't. It would be impractical. So we started working on a stochastic perfumer in order to scale the model to UK biobank size datasets. The main idea of a stochastic perfumer is to replace the classic variational inference with a stochastic variational inference, meaning that instead of visiting the entire population of the subjects per model iteration, we only visit a subset of subjects per model iteration. However, we don't want this randomization to compromise the accuracy of our results, so we take some extra things into account. Firstly, in order to be able to preserve a representative group model, we treat group level parameters as global parameters that keep a memory of not only current state, but also all the previous states that we have visited. Additionally, we want to make sure that the subjects remained aligned with the most updated group model, and therefore we use group level priors to estimate subjects. And finally, in order to allow for more heterogeneity in the population, we not only keep a memory of group level updates, but also subject-specific updates. By making these changes in the model, we were able to reduce the RAM requirements of the perfumer model quite significantly, so that we can use it to analyze 100,000 subjects in UK Biobank using the available compute servers. Right, now that we have the model, we obtained results from 5,000 subjects from UK Biobank. The data were minimally pre-processed for artifact rejection, smoothed, and then we characterized 150 RSNs at population and individual level. And the stochastic parameters included thousands of updates at group level, with each subject being updated 50 times on average. And here are all the RSNs. So they include both distributed default mode type networks as well as contiguous modes. And by getting these RSNs, we have a high dimensional soft parcellation of the brain. And to look into a bit more detail, here are seven RSNs that we found in the visual cortex, different hierarchies of visual cortex, the group model on the top, and 3 examples of those at the bottom for 2 example subjects. By visual comparison between the 2 subjects, we can see qualitative differences between them. The next example is 3 variants of default mode networks. And as we can see, on the left is the group level, and on the right, 2 example subjects. As we can see, the modes are largely overlapping, but they also have a lot of spatial detail differences between them. Okay, in the next step, now we wanted to compare stochastic perfumer's result to the more established spatial ICA group level and the spatial ICA plus dual regression subject level results. And for that purpose, we used F-cells melodic. At group level, when we compared the 2 models, we saw that for half of the modes, there was a clear one-on-one correspondence between stochastic perfumer and melodic results. But for the other half, we saw that each perfumer model corresponded to multiple ICA modes. This happens because as we go to higher dimensions, perfumer tends to keep the modes distributed and overlapping, while ICA tends to split them into smaller non-overlapping submodes. We also compared the 2 models at subject level, and here on the left is stochastic perfumer top group and bottom example subjects, and on the right melodic and melodic plus dual regression. By comparing these 2 against each other, we can see 2 key differences. Firstly, perfumer seems to be more successful in suppressing background noise, and secondly, because of explicit between subject variability estimations, it yields more meaningful cross-subject variability results. And here is another example of a mode, and again, we see the same story here in terms of noise. Okay, The final thing that we tested was that to see how each of these two models perform for predicting results of cognitive tests in UK Biobank. And for that purpose, we look at the full output of each model, as well as the spatial layout of the RSN separately and functional connectomes separately. We take 68 cognitive tests from UK Biobank and predict the outcome, and we use each RSN separately to do this prediction. And here are the results. So these on the left, these are the results of the full model, with all the elements combined. And as we can see, particularly for best performing modes and cognitive tests, Perfumo outperforms melodic dual regression. Perfumo is on the y-axis and dual regression on the x-axis. When we looked at each element, in terms of the spatial layout of the modes, again, Perfumo wins. But when we look at the functional connectome, melodic dual regression does a better job at predicting cognitive outcomes. And this is in line with the recent publications that I mentioned from Bisterbusch and Harrison in terms of interpretability of functional connectivity of the two models. And these are the examples of the best performing modes for predicting cognitive tests. Most of them are distributed, such as default mode network, but there are some also combined, such as this visual mode or this cerebellar mode. Okay, so to conclude, stochastic Perfumo allows us to infer individual and population RSNs from large data sets such as UK Biobank. Because of a stochastic inference, we can afford to model more heterogeneity in big data populations. When we compared the results to melodic and melodic dual regression, at group level, they were comparable. At subject level, Perfumo had better estimation of cross-subject variability. And finally, when we used the models to predict the results of cognitive tests, the spatial maps and the overall model of Perfumo performed better. The functional connectivity of the melodic dual regression performed better, but also there has been discussions around the interpretability of one versus the other. Okay, I'd like to thank my collaborators, and thank you very much for listening.